cell membranes as fluid mosaics. You've ever seen like mosaic stained glass windows kind of stuff? Or just like the concept of a mosaic in art class? A lot of people use that word to describe cell membranes, only instead of being just like solid, they are also fluid. So, the plasma membrane, jump straight in. Functionally, separates the inside of a cell from its surroundings. And also, controls inbound and outbound traffic. All right, inbound and outbound traffic. Sorry, my brain short circuited for a second. Okay, so this allows the concept of selective permeability. Selective permeability, which essentially means that the cell allows some substances to cross more easily than others. You will talk more about selective permeability in your hybrid assignment for tomorrow. In fact, it is all about selective permeability, so be on the lookout for that. And now we get into the actual fluid mosaic model. So, cell membranes. Are fluid mosaics of lipids and proteins. A little side note, with some important hi. So, some important carbohydrates. We talked about the extracellular matrix last week. Talked about the extracellular matrix last week and how those really important carbohydrates kind of come in and out of the cell. Carbohydrates are also sometimes embedded in the actual plasma membrane itself to just help keep things more stabilized. And we'll give an example a little bit later. Yeah. So generalized structure. Actually, these lipids, the most abundant lipids, so not all the lipids, but most abundant lipid is our old pal, the phospholipid. You've likely talked about phospholipids in past classes, the general structure, a bunch of little heads, bunch of little tails. arranged because of hydrophilic and hydrophobicness. We're just going to briefly touch on that before we get into the actual fluid mosaic model. So, phospholipids, I'll switch colors. Phospholipids are omnipathic. So, phospholipids amnipathic, sorry. Amphipathic, which means they have both hydrophilic and hydrophobic regions. So we've got our little phospholipid here. That's our little phospholipid. The head is hydrophilic, which is water loving. Hydrophilic head. Hydrophobic tail. So when they're arranged in a sheet, you've got all of the heads lined up and all of the tails on the inside. Now, membrane proteins can also be amphipathic. So the proteins also have hydrophilic sections and hydrophobic sections. Usually that has to do with the charges on the amino acids that make up that polypeptide, which we'll get into much more detail when we talk about protein synthesis. This amphipathicness maximizes contact of hydrophilic protein regions and water. So those hydrophilic parts want to maximize their time spent in water or aqueous environments. So those hydrophilic molecules will orient themselves on the outside of the cell, and the hydrophobic parts of the protein will kind of hide into the hydrophobic part of the membrane. So we've got our membrane protein. And it's in the cell membrane. This region right here would be hydrophobic, and this region up here would be hydrophilic. This concept of proteins being able to integrate into the cell membrane leads us to our fluid mosaic model. Which is essentially, let's see, that the membrane is a Mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. So that phospholipid structure isn't solid. It's not. It's more gelatinous than it is like structurally sound, which is why we need that cytoskeleton that we talked about last week to kind of create that structure for us, so that the plasma membrane acts more like a fluid than a solid, and those proteins bob around and move throughout it, kind of creating a almost stained glass effect, not quite, in that kind of liquid model. Now, that fluidity of membranes is an important factor that allows life to take place. So, fluidity. So, membranes, unlike the two-dimensional pictures that we often see, are not static sheets of molecules locked in place. Stuff's always moving, always on the go, floating around, bobbing around, moving around. They are held together by hydrophobic interactions. The only thing holding our cell membranes together is the fact that those hydrophobic tails don't want to associate with water. So the fact that our phospholipids are amphipathic is the reason why a single cell can exist. Let's see. Most lipids and some proteins can shift sideways in the shape or in the plane of the membrane, almost like just like walking through the hallway all nice and uniform and somebody throws an elbow. One of those plasma or one of those lipids can like veer sideways. So most lipids and some proteins can shift sideways in the plane of the membrane. Can shift sideways in the plane of the membrane. Like throwing elbows. It doesn't pop a hole in it. It doesn't let stuff leak out that normally wouldn't leak out. It just shuffles them around a bit. Very rarely, but it can happen. A lipid will flip-flop. Meaning if this is our lipid in question, it can go from there to there. It's just gonna flip sides. But there's so many packed in there that that space that it was in is gonna be taken up relatively quickly. But it can flip sides. What happens more regularly is that lipids switch positions side by side. So this is our membrane. It can go from that to that, where it was there, but it switched with this guy. On average, lipids move 2 micrometers per second, which is the size of, or the length of many bacterial cells. These guys move fast. So one lipid can be on the top of a plasma of a bacterial molecule, and in the next second can be at the bottom of that bacterial molecule. These guys are moving quickly. That's why it acts almost like a fluid. And because these lipids are moving all the time, proteins can drift. It's much slower, and some are anchored in place by the cytoskeleton. But if there's not, it can kind of move around and float throughout the surface of the cell. Odds are there's some sort of cytoskeletal element holding it in place, but it can kind of move around just a little bit. Temperature can influence fluidity. If it's colder, they are tightly packed. So think about bacon grease, right? When you cook bacon, when you just got done, it's a liquid, right? Just like an oil coating the bottom of the pan. But if you leave that there and don't clean it immediately, it'll kind of congeal into like a paste, almost. Plasma membranes behave kind of similarly. The colder they are, the more tightly packed those membranes are, so the more solid they are. But that's not good, because we need that fluidity to kind of keep our cells going. So in cells that are used to like colder environments, cholesterol molecules can get wedged between phospholipids. And affect fluidity. So if those cholesterol molecules are in there, those phospholipids can't pack as tightly together, so they don't solidify as much as they would if that cholesterol wasn't there. So cells that are adapted to colder environments have more of this cholesterol in order to kind of ensure that the fluid mosaic kind of remains in place. The idea that this cholesterol membrane happens in cold water environments helps us understand evolution a little bit more. There's more evidence for evolution when we look into a plasma membrane. So a little evolution connection. variations in membrane lipid compositions appear to be evolutionary adaptations. Just like our or just like regular adaptations work in our more traditional evolution experiments. So an organism developing a camouflage to avoid predation. The same thing can happen on a more cellular level. An example here is that fish in cold environments, so like deep sea fish, have a lot of unsaturated tails. To increase membrane fluidity. So our unsaturated fats have a lot of double bonds, like we talked about when we talked about our biological macromolecules. The difference between saturated and unsaturated fats is how many double bonds that exist in that fat. Unsaturated fats have a lot of those double bonds and a lot less hydrogen, which means they can't pack as tightly together. So that unsaturation, those increase in double bonds, is an adaptation. If a specific fish had a lot more of these double bonds by accident, it can live in a colder environment, reproduce more, and therefore its offspring would have more unsaturated lipids in its plasma membrane. So in our cell unit alone, we now have at least two connections to the concept of evolution. Our endosymbiotic theory with our chloroplasts and mitochondria, and the variation in plasma membrane composition. All right. Any questions about this? All right, cool. I'm gonna do a brief talk about membrane proteins and their functions, and if we have time, hit up some membrane carbohydrates. So it would not be a mosaic if we didn't talk about what makes it a mosaic. It's not all lipids. We talked about the fluid part, and now we're going to talk about the mosaic part. So membrane proteins and their functions. I'm going to abbreviate functions just so I have space. Membrane proteins and their functions. There are two types of membrane proteins. The first type are integral proteins. Integral proteins penetrate that penetrate hydrophobic regions. Actually, hydrophobic interior of the plasma membrane. Many are transmembrane proteins, which means they span the entire membrane, like the one I draw earlier. Transmembrane proteins span the entire membrane. Some only extend partially. So they still go into the plasma membrane, but they don't go all the way through. The other type of membrane proteins are called peripheral proteins. Peripheral proteins aren't embedded, but they are loosely bound to the surface. Yeah. Not embedded. Loosely bound. This could be via lipid, this could be via carbohydrate, this could be via those extracellular matrix components that we talked about last week. So there are transmembrane proteins and there are peripheral proteins, or integral proteins and peripheral proteins. I'm going to talk about their functions. I've got 1, 2, 3, 4, 5, 6 different functions. There are probably a lot more, but these 6 different categories hit most of them. So we've got membrane protein functions. Membrane protein functions. Number 1, transport. You will talk a lot about these transport proteins in your selective permeability assignment tomorrow. So those are like hydrophilic channels and pumps. Stuff that transports things into and out of the cell. They can also help with enzymatic activity or have enzymatic functions. Goodness gracious. Or complete steps in metabolic pathways. When we talk about metabolism after we finish up with the cell, we will talk a lot about these membrane proteins that have enzymatic activity. They're not in the outer membrane as much as they are in the mitochondria and chloroplast membranes, but they do exist on the outside of the cell as well. Third function is signal transduction. So receptors and signal releases. If you think about neurotransmitters on your neuron brain cells, right? At the end, we've got little stuff or little receptors that spit out our neurotransmitters, and another cell has the receptors to recognize that signal and continue it on. That is due to our membrane proteins. Number 4 is cell-to-cell recognition. So proteins that act as ID tags. That's like the antibodies on the outside of your blood cell that tell you whether or not you have A, B, O, positive, negative blood. Those are all proteins that exist on the outside of a cell that help any organism, I guess, recognize that cell as self or as foreign, so the immune system can attack it if it is foreign. Also, intercellular joining. We talked about these with our tight and gap junctions. Those tight junctions are just proteins that hold cells really, really closely together. And the last function, oh, that is 6, not 5, is attachment to the cytoskeleton. And this helps us maintain shape. Those integrin proteins, or the fibronectin, I guess, help us connect to our cytoskeletal, integrin proteins we talked about with the extracellular matrix are our protein spanning membranes that connect to the cytoskeleton on the inside and the extracellular matrix on the outside. Those are examples of how proteins can attach to the cytoskeleton and help us maintain shape. So, membrane proteins do a lot of stuff. Let's see. Yeah, I got time. We're going to talk about membrane carbohydrates. Real quick. Nothing too crazy. Membrane carbohydrates. See, they are really good for cell-to-cell recognition. This is really good for sorting cells into tissues. In development. So when an embryo is forming and developing, different cells are bound to become different parts of the body, different tissues, and the carbohydrates can help the organism determine which cells are going to stay in which part of the body. Also really good for immune system stuff. Helping recognize cells as self. If a foreign carbohydrate exists on a cell, immune system can be like, not us, attack, and so on. They are structurally short, branched, and covalently bonded to lipids. And this creates glycolipids. Just like when we had carbohydrates bound to proteins, we had glycoproteins. Lipids bound to carbohydrates are glycolipids. All right. One last little tidbit I forgot, and that is synthesis of sidedness. Cells don't look the same on the inside as they do on the outside. Like we said, those proteins, sometimes they only go halfway through the cell. How do we know which side of the cell or which side of the plasma membrane is going to be the inside and which side is going to be the outside? Because they do have distinct inside and outside faces. I can't spell. Distinct inside and outside. And that is determined, that asymmetry is determined as the membrane is built in the ER and Golgi. So as that Golgi apparatus, as that endoplasmic reticulum are building our plasma membranes, they determine which cell is the inside and which part is the outside. When you get that done, about 4 minutes left. There is an exit ticket on Canvas. You can knock that out for me, please.